Good morning, students. Welcome back to Botany class. In previous class, I started the chapter Biological Classification. So in biological classification, I explained you about the two kingdom system of classification, three kingdom system of classification, four kingdom system of classification. After that, I started the five kingdom system of classification. Two kingdom system of classification proposed by the scientist Carolus Linnaeus. He classified, he proposed two kingdom system of classification. So there are two kingdoms, one is kingdom Plantae and another one is the kingdom Animalia. So in kingdom Plantae, he has included bacteria, cyanobacteria, as well as the fungi, mosses, ferns, gymnosperm and angiosperm. So these he included under the kingdom Plantae. In kingdom Animalia, protozoa, invertebrates and vertebrates. Here we can observe that actually bacteria, cyanobacteria, these are prokaryote. So scientist Carolus Linnaeus, he did not distinguish prokaryote and eukaryote. Second one, he did not differentiate between the photosynthetic and non-photosynthetic. Algae are photosynthetic, it synthesizes its own food material. But fungi are the heterotroph. And second one is cell wall. When it comes to cell wall composition, algal cell wall or commonly plant cell wall is made up of cellulose, but fungal cell wall is made up of chitin. But scientist Carolus Linnaeus, he included these all into a group kingdom Plantae. So these are the some drawbacks. What are the drawbacks? He did not distinguish prokaryote and eukaryote. Second one is the a very important one here we can be observed that cell wall composition is also different than to be has added in that. So due to this drawback, two kingdom system of classification is not be accepted. After that, three kingdom system of classification which was proposed by the scientist Ernst Haeckel. Scientist Ernst Haeckel, he increased which kingdom? He increased the kingdom Protista. So all unicellular and eukaryotes included under the kingdom Protista. Then scientist Copeland, he proposed the four kingdom system of classification. So he increased a kingdom, which kingdom? Kingdom Monera. So there were four kingdoms. One is kingdom Monera, second one Protista, third one Plantae and fourth one is the Animalia. After the four kingdom classification, scientist R.H. Whitaker, he proposed five kingdom system of classification. So what are those kingdom? Kingdom Plant, kingdom Monera, second one is the Protista. Scientist R.H. Whitaker, he increased which kingdom? He increased the kingdom fungi, or it is also called as mycota. So according to Whitaker, there are kingdom Monera, Protista, fungi, Plantae, and last one is the Animalia. Scientist R.H. Whitaker, he used some criteria. In yesterday's class, I explained you one criteria he used for five kingdom system of classification. First one is the cell type, whether the cell is a prokaryote or it is a eukaryote. All prokaryotes and unicellular, Prokaryotes and unicellular organisms included in which kingdom? Included in kingdom Monera. Included in which kingdom? Included in kingdom Monera. And remaining other unicellular and eukaryote he has included under the kingdom Protista. And in fungi, multicellular heterotroph with cell wall, those organisms included under the kingdom fungi. Then all autotroph included under the kingdom Plantae, and all heterotroph organisms included under Animalia. So it means that the first character or criteria he used for system of classification is cell type. Okay, so that in previous class same I started, that one is cell type, prokaryote or eukaryote. Second criteria, what criteria he used? Second criteria, body organization, whether the organism body is made up of single cell or the organism body is made up of many cells. If organism body is made up of single cell, what we'll call unicellular. If organism body is made up of many cells, what we'll call multicellular. So second criteria used, that one. A third criteria is thallus organization. Thallus organization, plant body is differentiated or body is not differentiated. So this is the third criteria he used. And the fourth one is the mode of nutrition, whether they are autotroph or heterotroph. Is it clear? Whether organism synthesizes its own food material or the organism depends upon other organism for food. So this is the fourth criteria. Then reproduction. Formation of new individual with fusion or with formation and fusion of gametes, or parent directly gives rise to new individual. Whether reproduction is sexual or maybe sexual. Last one, phylogenetic, means evolutionary relationship. So these are the criteria. Very important it is. What are the criteria? Cell type, body organization, thallus organization, mode of nutrition, reproduction, and the last one is the phylogenetic. So these are the criteria used for the five kingdom system of classification. Because in yesterday's class I told that bacteria, cyanobacteria included, along with bacteria, cyanobacteria, fungi also included under the kingdom Plantae. But scientist R.H. Whittaker, he separated all these. Bacteria, cyanobacteria included in kingdom Monera. And fungi, he made separate kingdom, that is fungi kingdom, fungi, or it is also called as the mycota. All plants belong included under the kingdom Plantae, and all Animalia included under the kingdom Animalia. And even amoeba and paramecium. Amoeba and paramecium earlier placed in kingdom Animalia, but later amoeba and paramecium placed in a kingdom Protista. Is it clear? So after that, in previous class, I started characteristics of five kingdom system of classification. So what are the characteristics? Yesterday, just I wrote the characteristics. So once again, just you have to see the what are the characteristics of five kingdoms. It's a very important. First character, cell type. So in previous class, yesterday I already told that cell type, monerans are prokaryote, remaining all are eukaryote. Here, only monerans are prokaryote, remaining all are eukaryote. Moving to the cell wall. Cell wall present but non-cellulosic. Non-cellulosic. Okay. Second one is here the protista present in some. Present in some. Is it clear? In the member of fungi or mycota? Present. But non-cellulosic. Yesterday class I told that fungal cell wall is made up of chitin. It is made up of chitin. Made up of chitin. Okay. Then plantae cellulosic. And animalia cell wall is absent. Is it clear? So cell wall in Monera, cell wall is present but non-cellulosic. It means that in the member of the bacteria, in Monera, the cell wall is made up of, it is made up of the polysaccharide. Polysaccharide plus amino acid. Amino acid. Is it clear? In the member of the protista, cell wall present in some. It means that it is absent in some. Fungi present but non-cellulosic, made up of chitin. Plantae, cell wall present and made up of cellulose. In animalia, cell wall is completely absent. Okay, clear? Moving to the third character, that is the nuclear membrane. Nuclear membrane. Third character is the nuclear membrane. Nuclear membrane. In Monera, nuclear membrane is present or absent? In Monera, nuclear membrane is absent. It is absent. In the member of the protista, present. In mycota or fungi, present. In plantae, present. In animalia, present. Animalia also present. It means that nuclear membrane is absent in Monera, present in protista, fungi, plantae and animal. Moving to the next character, that is the body organization. It is already I told yesterday that body organization in Monera, cellular, protista, cellular, fungi, multicellular, loose tissue and plantae tissue as well as the organ. In animalia, tissue, organ and organ system. Last character, mode of nutrition. In the member of the Monera, the mode of nutrition is autotroph. It may be the photosynthetic autotroph or the chemosynthetic autotroph or heterotroph. Heterotroph, either parasite or saprophyte. Saprophyte. Is it clear? In the member of the Monera, the mode of nutrition is autotroph. It may be the photosynthetic or chemosynthetic, or heterotroph. It may be the parasite or it may be the saprophyte. Protista. Coming to the Protista. In Protista, the mode of nutrition is autotroph, that is the photosynthetic, and second one is the heterotroph. It means that in Protista, we can observe both autotroph as well as the heterotroph mode of nutrition. Moving to the third one, fungi. In fungi, purely they are heterotroph. They do not synthesize their own food material, depends upon other organism for food. So they are purely heterotroph. Moving to the next one, plantae. In plantae, you know very well, they are autotroph, photosynthetic, photoautotroph. And in Animalia, the mode of nutrition is heterotroph or holozoic or saprophytes. Holozoic means here the inside the organism body digestion process is taking place, that is called as the holozoic. Saprophytes means they consuming the food from dead and decaying organic matter. Clear? Saprophytes means they consuming the food from dead and decaying organic matter. Is it clear? Monera also heterotroph, parasite or saprophyte. Parasite means the organism consuming the food from host, that is called parasite. Saprophyte consuming the food from dead and decaying organic matter. So these are the characteristics of 5 kingdoms. Clear? So after this we will go to the next one. So this completes the characteristics of 5 kingdoms. Okay, after this, after the 5 kingdoms, over time, attempt has been made to evolve classification system which not only reflects morphological concentrate, very important point it is. Over time, attempts have been made to evolve classification system not only reflects morphological, physiological, reproductive similarities, but it also reflects the phylogenetic relationship. That is the what? Evolutionary relationship. In this chapter, we will study kingdom Monera, kingdom Protista, kingdom Mycota of Whittaker classification. We will study Plantae and Animalia, but in brief. Monera, Protista and Mycota we are going to study in detail, but Plantae and Animalia we are going to study in brief. So after this I will start the first kingdom, that is the Monera. Moving to the first kingdom, that is the kingdom Monera. What are the characters of kingdom Monera? Concentrate. First character of kingdom Monera. What is the first character of kingdom Monera? Monerans are primitive, prokaryotic and unicellular. Understanding here? So they are the primitive. They are the first formed organisms on earth. Second one, prokaryotic. In yesterday's class, I told you that what is meant by prokaryotic. What is meant by the prokaryotic? Prokaryotic means just you take a one monomer cell. This is the cell wall. This is the genetic material that is commonly, which is the genetic material? DNA. This region we are going to call the cytoplasm. Okay, here we are going to observe. This is the plasma membrane. Here we are going to observe, this region is called as the cytoplasm. In this region we are going to observe the ribosomes. Prokaryotic cell means genetic material is present. Genetic material is present, but this genetic material is not surrounded by any membrane. So, such type of cell is called as the prokaryotic cell. Understood? So, in previous class, yesterday's class, I told you what is a prokaryotic cell and what is a eukaryotic cell. So, monerans are prokaryotic and organism body is made up of single cell, so that is the unicellular. Monerans are primitive, prokaryotic and unicellular. Moving to the second character, of the monerans. Second character of monerans, nuclear membrane and nucleolus absent. Nuclear membrane and nucleolus absent. What is meant by, you know very well, nuclear membrane? What is meant by the nuclear membrane? Genetic material is surrounded by a membrane called as the nuclear membrane. But in Monera, nuclear membrane is absent. Genetic material is present, but nuclear membrane is absent. Then, what is meant by the nucleolus? Nucleolus means it is present in eukaryote. So, inside the nuclear membrane, a dark spherical structure called nucleolus. This is called as nucleolus. A dark spherical structure found inside the nucleus, or we can tell inside the membrane, is called as the nucleolus, and that nucleolus is absent in where? Monera. So what is the second character of the monera? Nuclear membrane and nucleolus absent. Moving to the next character, that is the third character of monerans. Third character. Third character is lack cytoplasmic cell organelles. What are the cytoplasmic cell organelles? What are the cytoplasmic cell organelles? Cytoplasmic cell organelles such as mitochondria, endoplasmic reticulum, Golgi complex, lysosome, plastids. These cell organelles are completely absent except ribosomes. Is it clear? So, monerans lack cytoplasmic cell organelles except ribosomes. It means that ribosomes are completely absent. Completely absent. These cell organelles are not present. What are those cell organelles? Mitochondria, endoplasmic reticulum, Golgi complex, lysosome and plastid. These are completely absent. Okay. Moving to the next character. Next character of Monera, DNA is not associated with histone protein. DNA is not associated with histone protein. Now question is here, what is histone protein? What is meant by the histone protein? It is a basic protein. Histone is a basic protein rich in two amino acids. One is lysine and another one is the arginine. In the member of the Monera, DNA is present, protein is also present, but that protein is not histone protein. DNA is not associated with histone protein. Is it clear? But it is associated with, we can tell that, other protein. Okay, so this is a very important character. Next character. Next character is the mode of nutrition is autotroph or heterotroph. Heterotroph may be parasite, or saprophyte. Parasite, I told that organism consuming the food from host. For example, you have to take down the example is the typhoid. This is a common disease we are going to observe in human. That which bacteria? Typhoid is caused by a bacteria Salmonella typhi. Salmonella typhi is a parasite. It means that it consuming the food from host or the living. So heterotroph may be parasite. Second one is the saprophyte. Saprophyte, they consuming the food from dead and decaying organic matter. Is it clear? Moving to the next character is a very important character of kingdom Monera. Ribosomes are 70S type. There are two types of ribosomes. One is 70S and another one is the 80S. 80S ribosome commonly we are going to observe in eukaryote. We are going to observe in eukaryote. S indicate. What is the S? S means here on the basis of sedimentation coefficient. Sedimentation coefficient. On the basis of sedimentation coefficient, ribosomes classified into two types, 70S, 80S. In Monera, ribosomes are 70S. In eukaryote, 80S. On what basis they classify? On the basis of sedimentation coefficient. This is a very important character of the Monera. 70th type of ribosome only we can be observed in prokaryote. In eukaryote, 80S, but in some of the organelles, we are going to observe the 70S type of ribosome, such as in mitochondria and chloroplast. We are going to observe the 70S type of ribosome. Is it clear? Moving to the next one, next character, asexual reproduction. Asexual reproduction takes place by binary fission, last character of the Monera. Asexual reproduction takes place by binary fission. It is one of the most common type of asexual reproduction that we are going to observe in bacteria. And in some other we are going to observe the spore formation. So it means that asexual reproduction takes place by binary fission and spore formation. But the most common is binary fission. What is the binary fission? A single parent cell binary fission means bi means two. Parent cell undergoes division to form two daughter cells. One parent cell gives rise to how many daughter cell? Two daughter cells. Again, this one mature and gives rise to how many daughter cell? Two daughter cell. So this type of asexual reproduction is called as the binary fission. Spore formation means these are the minute microscopic structure. They germinate and gives rise to new individual. And sexual reproduction is primitive type. Sexual reproduction is, so these are the characters of kingdom Monera. What are the characters of kingdom Monera? Moneras are primitive, prokaryotic and unicellular. Second one is nuclear membrane and nucleolus absent. Third character, lack cytoplasmic cell organelles such as mitochondria, Golgi complex, lysosome, as well as plastid. So these are absent in the monerans. Then mode of DNA is not associated with histone protein. Genetic material DNA is present, but it is not associated with histone protein. Moving to the next one, mode of nutrition is autotroph. It may be photosynthetic or chemosynthetic autotroph or heterotroph. It may be parasite or saprophyte. Moving to the next character, ribosomes. Other cell organelles are all organelles are absent except ribosomes, and here ribosomes are 70 years type. Last character, reproduction takes place by asexual and sexual. Asexual reproduction takes place by binary fission and spore formation, and sexual reproduction is primitive type. So these are the general characteristic features of the kingdom Monera. Next one is here, Monera. It includes next one, Monera. It includes Monera. It includes bacteria, cyanobacteria, archibacteria, and mycoplasma. Okay, is it clear? So Monera includes the bacteria. Bacteria belongs to which kingdom? Monera. Cyanobacteria belongs to which kingdom? Monera. Archibacteria belongs to Monera. Mycoplasma belongs to Monera. So now we will study first. We will study first one, bacteria. Bacteria are the sole members of the Monera. Sole means, meaning is sole means here the word single. Is it clear? Bacteria are the sole members of sole members of they are the sole members of the kingdom Monera. Bacteria, they are bacteria are minute, very very small they are. They are microscopic, present everywhere. Understanding here? Bacteria are very minute, microscopic and present everywhere. Everywhere means they are present in soil, they are present in water, they are present in air, even they are present in the extreme habitat such as they live in hot spring, geyser, and they live even in ocean. So it means that they are present everywhere. They are present in soil, they are present in air, they are present in water, they are present in hot spring, as well as they are present in ocean. Hundreds of bacteria are present in handful of soil. If you take a small amount of soil, in that we are going to absorb number of bacteria. So they said that hundreds of bacteria are present in handful of soil. Okay, then Moving to the next one is here the shapes. Bacteria classified into four types. Bacteria classified into four types on the basis of shape. They classified how many types? Four types. So what are those four types of bacteria? Four different types of bacteria classified on the basis of shape. What are those? There are four different types of bacteria classified on the basis of shape. First shape, that is the cocci. Second one, bacillus. Third one, vibrio, and fourth one is the spirula. What is cocci? Spherical shaped bacteria is called cocci. Is it clear? Spherical shaped bacteria is called as the cocci. Is it clear? Second one, bacillus. What is meant by the bacillus? Rod-shaped bacteria is called bacillus. Rod-shaped bacteria is called bacillus. Third type is vibrio. Comma-shaped bacteria is called as vibrio. Comma-shaped bacteria is called as vibrio. Next one, spirula. Spiral-shaped bacteria is called as the spirula. So, these are the four different shapes of bacteria. What are those? Cocci. Cocci means spherical shaped bacteria is called as cocci. Second type is bacillus. Bacillus means rod-shaped bacteria is called as bacillus. Third one, vibrio. Vibrio means comma-shaped bacteria is called as vibrio. And last is the spirulum. Spiral-shaped bacteria is called as the spirulum. So these are the four shapes of bacteria. Even cocci, if only one spherical structure, what we are going to call? Monococcus. If, like this structure, this is called as what? Monococcus. If they are occur in tail, what will call? What will call? Monococcus. If two are there, then we are going to call as the diplococcus. If four, tetracoccus. If they are occurring chain like this. If they are occurring chain, what we are going to call? That is called as the streptococcus. Streptococcus. Is it clear? If one, we are going to call one single spherical structure called as the monococcus. If they are occurring pair, then we are going to call as the diplococcus. If four, we are going to call as a tetracoccus. If they are occurring in this like chain, then we are going to call as the streptococcus. It is a very important. So bacteria classified into four types on the basis of shapes. How many shapes? Four. What are those? Cocci. It is a which shape? Spherical. Second one, bacillus, rod shape. Vibrio, comma shape. Spirulum, spiral shape. So this is about the types of bacteria on the basis of shape. Moving to the next one, we'll come to the bacteria. Like in general characters of Monera, I told that they are unicellular, they are primitive, they are the prokaryote. So it means that bacteria also unicellular. Okay. Second one is we are going to study in detail about the bacteria. They are unicellular, they are primitive, they are prokaryote. Okay, mode of nutrition is autotroph or heterotroph. Okay, these all already I told in general characteristics of Monera. Coming to the next one, bacteria. In bacteria, cell wall is present and cell wall is made up of peptidoglycan. It's made up of what? Peptidoglycan. Peptidoglycan, it consists two parts. One is the peptide and another one is the glycan. Peptide means amino acid. Is it clear? And glycan means sugar. It means that bacterial cell wall is made up of peptidoglycan. It is not made up of cellulose. It is made up of peptidoglycan. Next one. At one side, just I will draw the diagram of the bacterial cell, ultrastructure of bacterial cell. Just we have to see this is the cell wall. This is the just you have to see this is the ultrastructure of bacteria. Through this diagram, I will be explaining you about what are the parts of the bacteria. So, in bacteria, cell wall is present. This is the cell wall, and cell wall is made up of peptidoglycan. It means that it is a combination of the amino acid and sugar. Next one is here. Cell wall is surrounded by thick or thin layer. Cell wall is surrounded by thick or thin layer. So that layer is called as glycocalyx. That is called as the glycocalyx. If the cell wall is surrounded by thin layer, it is called slime layer. If it is surrounded by thick layer, called capsule. If bacterial cell wall is surrounded by thin layer, called as slime layer. If it is surrounded by thick layer, called as the capsule. So in next class, I will be tell you about what is the chemical composition of the capsule or the other parts of the bacterial cell. Is it clear? Thank you, students.